Hi, everybody. I'm Sani, and together with my fellow student associate Grace, we've been working together with Dr. James Tono's group from Biomedical Engineering on advancing cervical cancer diagnostic technology. From the feedback we received from reviewers, we focused on effort on two things: path to market and partnership development for our laser microbiopsy technology. We worked alongside our mentors, Rosemary French, Hector Torres, and Jessica Calderon Mora, who gave us invaluable insight on all facets of our product to be translated into the point of care setting. We're passionate about improving women's healthcare accessibility, and this experience has shown us how to bridge engineering innovation with real clinical needs. Our team has made exciting progress on prototype development, and we're now exploring strategic partnerships to advance towards clinical testing. I'll now hand it over to Alex to share more about technology, so please welcome him to the stage. Thank you.
All right, hi everyone. So diving right in here, after a patient initially receives a positive screen from a pap smear or HPV test, they're brought in for a colposcopy. This is a careful examination of the cervix, but is highly subjective depending on the provider's experience. During that procedure, if there's an area of interest that's noted, or perhaps at a subsequent procedure, a biopsy is required for diagnosis. Biopsies are very painful, and this pain often limits the number of samples that a provider can actually collect. After that, the biopsy is analyzed and the appropriate therapy is recommended, at which point the patient has to come back in for a follow-up procedure. During this entire process, there are many additional follow-ups that are required, and as a result, many patients get lost in the cracks of treatment. Our solution from MicroBX aims to solve this problem by essentially compressing colposcopy, biopsy, and diagnosis into one procedure. This would limit the number of patients that are lost to follow-up and also addresses the pain aspect of biopsy collection. Overall, this would contribute to an over $1 billion savings opportunity as a result of earlier intervention and accessibility for patients. Our system utilizes a novel donut-shaped beam that essentially extracts a small fragment of tissue on the order of a few hundred microns. This tissue is then analyzed by our point-of-care analyzer in order to provide immediate feedback to the physician and guide subsequent therapy. Compared to existing technologies, our product is way less traumatic and painful for patients, but still offers the same diagnostic power. So from our reviewers, we were advised to follow a phased approach to development, where phase 1 primarily focuses on our harvesting device, which would likely follow a 510(k) pathway and could be integrated into existing workflows. Next, phase 2 would involve our diagnostic analyzer, which would also end up working with some sort of strategic partner for development and would likely follow a PMA. This would be the aspect that completely disrupts the existing protocol. Overall, this approach de-risks development and the release of our device. So this is a very high-level look at our timeline here. The main things I want you to know are that our technology is already patent pending, and we recently received the Texas POC award. However, we require additional funds to really get our technology at a clinical stage. We have a strong technical team featuring clinical advisors from Dell Medical School, and I will leave it here at our value proposition. I'm happy to take any questions.
Great, thank you, Alex.
Nice presentation, Alex. I was just wondering, do you have any evidence that the sensitivity and specificity of this approach matches or surpasses the current standard of care?
The idea is that we're able to collect more biopsy samples per patient, which is the limiting factor right now. So the reason why sensitivity is so low is that on average, only one biopsy is collected per colposcopy. And studies seem to generally show that at least having 3 or more biopsy samples contributes to an over 95% sensitivity. So we're able to lower the bar for basically collecting more biopsies.
Thank you. Great presentation. So does your biopsy system have to use your analytical system as well, or is it agnostic with current platforms?
This is the nice part. So our first harvesting tool can be integrated into existing workflows. So that sample can be processed similarly in a pathology lab like they do now. But there's technologies already out there, and we've already demonstrated in our lab that we can create a device that basically bypasses the traditional pathology lab and provides immediate feedback to the physician.
So then is there a reason that because it's a PMA, it's going to require significant investment. So is there a reason that you're also investing in developing the analytical platform as well?
This is particularly why we're looking at strategic partnerships, so we don't have to take on as much risk with that development.
Alex, it's a little unclear to me what the advantage of point of care performance of the biopsy specimen.
Essentially because the patient doesn't have to come back. So particularly for underserved and rural communities, patients just simply don't come back in for follow-ups. And so even if they're diagnosed, they just don't come in to get the appropriate therapy, which is what contributes to the very high rate of preventable late-stage cervical cancer.
Thank you.
So we can basically get them while they're already in the office.
Great. Thank you, Alex.
Thanks.
Yeah, the next group, please.